For the next 20 minutes or so, we're going to take a little bit of a detour, probably from the kind of conversation I think we've had for most of the day already. I'm not an expert in complex in vitro models, so I'm not going to pretend to speak about them. But rather, when asked to speak, I was going to give an overview of how some other technology is translated into sort of more routine use at FDA. So I'll be talking a little bit more about precision medicine and how that's evolved over the past 15 to 20 years. So as mentioned, I'm in the Office of Clinical Pharmacology. I'm a clinical pharmacologist. I'm a pharmacist by training. I work in the Division of Translational and Precision Medicine. So we focus mostly on precision medicine, pharmacogenetics, as well as a lot of rare disease work where we're looking at translational models to figure out evidence of effectiveness, dose, and those kinds of issues in specific drug development programs. So I'll talk during this session a little bit about how translations have been innovations have been translated into drug development and practice to some extent. I will go over a case study that's not a complex in vitro model, but rather a simple one, and then talk about some other opportunities for these types of complex in vitro models in drug development. So just to level set a little bit, precision medicine, as a clinical pharmacologist and a clinical pharmacist is basically everything, we do day in and day out. We try to identify factors that might warrant changing a dose for a given patient or excluding a certain patient from treatment based on organ function or drug interactions and those sorts of things. So really everything we do is precision medicine in a way. The way this has been talked about for many years is really taking a biomarker and testing a patient for it and then matching the drug to that person's pathobiology to some extent. And this is obviously most well understood in the setting of cancer. So these biomarkers typically would identify patients who are eligible for treatment, as is the case with most targeted cancer therapies, would allow for aiding and determining dosages, such as drug metabolizing enzyme polymorphisms that might warrant smaller doses for some people, or dictate how intensively someone might need to monitor response to individualize the therapy. This could be a dynamic biomarker, it could be a predictive biomarker that would potentially increase the risk of a safety event, for example. So this is a very busy slide, and I'll probably dwell here for a little bit just to kind of walk through the history. And if I get through nothing else in the time allotted, that's fine, I think. So precision medicine in modern terms really dates back to 1998 with the approval of trastuzumab for HER2 positive cancer. This was kind of the sentinel event, but it wasn't for several years that I think FDA really made a commitment with Janet Woodcock kind of verbalizing interest in advancing precision medicine and personalized medicine through pharmacogenetics. Later on, the Human Genome Project was completed, so we obviously had a wealth of data that was available at that point in time. And as companies were trying to negotiate how to work precision medicine into their drug development programs, there were workshops on pharmacogenetics. So there was open public discussion about how to advance these types of technologies in practice. And at some of those early workshops, there was discussion of the safe harbor concept. And what that ultimately led to was the creation of the voluntary genomic data submission program at the time. Later it was called the voluntary exploratory data submission program. And that was a really important milestone, I think, in FDA's advancement of these types of technologies. Basically, the voluntary data submission program over the course of several years allowed industry to interact with FDA in a non-regulatory capacity. So we were able to talk to companies about what their challenges were with respect to data generation. We were able to have hands-on experience with submission of data to FDA that might have been complex, like such as data from chips or large volumes of data. So a lot of those kinds of things happened in the course of those data submissions and meetings with companies. This has now kind of been sunsetted and hasn't really been active for many years at this point. The critical path innovation meetings, which some of you may be familiar with, have somewhat replaced this. At that time, after we had gotten some experience, put out guidance on how to submit data. So there was some initial policy that came out of some of those early interactions. Around the same time, the biomarker qualification program was put into place. Our staff who have expertise in pharmacogenetics were becoming more integrated into the regulatory review of marketing applications and IND submissions. So it became much more commonplace for FDA to have a more proactive stance in looking at these programs and identifying opportunities for personalization so those types of studies could be done. What then followed was KRAS testing for the anti-EGFR drugs. This was a pretty significant milestone also because there was a lot of debate about taking retrospectively generated data and transforming the indication statement for a drug. So there was advisory committees held, and ultimately those drugs were narrowed in terms of their indication to specific subpopulations that were expected to respond. With that experience, I think and seeing a lot of drug development programs were heading in this direction, we're able to put together guidance to tell companies when certain types of genomic studies would need to be conducted, as well as what policies were around co-development of companion diagnostic tests. As time went on, this was a few years later, PDUFA 5 negotiations came into the fore, and there were enhancements under PDUFA 5 for biomarkers and pharmacogenetics, which also gave some additional resourcing and interest in this area to kind of make sure FDA was ready to review, had the resources to look at these kinds of issues, as well as clarify the regulatory pathways. What followed from there was a couple of other guidances, trial enrichment, co-development. But as this was going on, we started seeing a very large number of drugs being approved for molecular subsets of patients, specifically in lung cancer. There was a number of drugs approved early on, crizotinib for ALK-positive lung cancers and a variety of others. And then what happened after that is they just got more complex. So we started seeing panels of tests being performed, homologous recombination repair deficiency being one for breast and other types of cancers. So we started seeing complications of the co-development programs. And there's also, there's a couple of things going in parallel here. So there's like targeted drug development, and then sort of the studies that facilitate that, like genomic sampling and clinical trials, which is essential to be able to do genomic studies in a drug development program. So we worked with our international regulators to develop the sampling guidelines under ICH E18. So there was more of a global effort. And then what followed up until present day is really just more complications on the same issue. How do we deal with patient subsets that are very rare? What type of evidence do you need? Because you can't really obtain clinical trial evidence. We have genetically targeted therapies such as the antisense oligonucleotides becoming more commonly developed. Those presented new and unique challenges. Started seeing tissue agnostic approvals in cancer, where drugs are not targeted to a specific cancer type, but rather molecular alteration. And then now we have a number of individualized therapies, or these antisense oligos that are being designed for individual patients. So you can see from this that over time there was a growing level of comfort, and along with that, a lot of policy and infrastructure development that went into sort of making this now rather routine part of drug development. So you can see over the past 10 or so years, I don't know what color this is because I'm somewhat colorblind, but the targeted therapies line is the one I'm interested in. This is drug approvals as well as some other product attributes. But you can see targeted therapies have steadily occupied about 20 to 30% of the drug approvals over the past decade or so. So clearly this is a mainstream thing now, whereas 10, 15, 20 years ago, it was an issue that was highly complicated and nobody really knew how to navigate the pathway. And I think what the keys to this were, as I somewhat alluded to, we built regulatory science capacity, we built regulatory review capacity and expertise, developed policy, worked with our stakeholders to understand what the issues were, launched pilot programs, and really communicated to the extent we possibly could what our current state of thinking was. So that's sort of how things evolved. And if you look at other programs, like now we have the Model Informed Drug Development Program, which started under PDUFA 6, it follows pretty much a similar trajectory from exploratory and not knowing how to handle it to now becoming somewhat more routine. So I'll walk quickly through a case study, specifically the targeted therapies for cystic fibrosis. So we have a number of drugs now approved for cystic fibrosis. If you're not familiar with cystic fibrosis, there's a number of different mutations that each have different kinds of effects on the channel. It's basically a channelopathy. It's a lung disease, very disabling, results in premature mortality. About 10, 15 years ago, drugs were starting to be developed that actually targeted the channel. So they were disease modifying potentially. The first of which was ivacaftor, which basically facilitates chloride transport by potentiating the open channel probability of the CFTR protein, which is what's the variant protein in this condition. And that was the first one to come in, and we'll talk a little bit more about that approval. Lumacaftor, Lexicaftor, Tezacaftor were all other, all the subtly different mechanisms that were able to target different populations. So Ivacaftor, in some of their initial preclinical work, basically found that in Fischer rat thyroid cells and human bronchoepithelial cells, that only some of the variants were responsive, specifically the class 2 variants that affected the open channel probability at the cell surface. And you can see on the right-hand side some of the data for the different variants and what happens when you add different amounts of drug. There's some variability. So basically what they decided to do at that point was target their development program to patients with a specific variant, the G551D variant. And that was what their initial clinical trials were. And they basically showed a clinical benefit, which is shown in the bottom panel, in patients who had this specific variant. And then they did another variant they studied another variant that's the most common variant in cystic fibrosis at 508 deletion, and it showed that there was no effect. So clearly driving home that proof of concept. So we have, this in vitro model. We have clinical trials that demonstrated the translation of that model to clinical benefits. They then went on to expand the clinical trial population. So there's some additional variants that were found to be responsive in vitro. So they did another clinical trial with some of these additional variants all mixed together. Again, showed response. Indication was expanded at that point in time. And then ultimately the company came and basically wanted to be able to allow the indication to be expanded to basically any patients that had a response in vitro, patients with a variant that was responsive in vitro. So after much, discussion and evaluation of the data, ultimately it was approved for 23 additional responsive variants. And then that was basically written into the indication, which I'll show on the next slide, that patients with an in vitro responsive variant are able to get the drug. And then this also then paved the way for the development of various combination products and other drugs in this that target this channel. So the first version of the labeling that had this expanded indication shown on the left-hand side, basically you can see the in vitro effect relative to the different variants that were being studied. And as time went on, as I mentioned, we expanded it to be approved for any patients who were shown to have a variant that was responsive in vitro. And now it's basically distilled down into a table, and there's not clinical data necessary to justify use in any additional patient populations. So what fed into all of this was really, that we had a robust and pretty well validated analytical and assay system. We had a pretty good understanding of the channels and what the variants did to the channel's functionality. We were able to verify the data internally. So the company submitted raw data to us. We evaluated it. And then we had some experience with the clinical trials showing that those findings in vitro were portable to the clinical experience. So to the right is a kind of a list of questions we had posed. The 2 publications on the bottom, you're free to go read. These are sort of outlined there, but covers a lot of the issues that we were concerned with. So understanding of the variants, is the mechanism of the drug well understood? Do we understand the disease and so on? So that's not a complex in vitro model. It's a simple model, but nonetheless, it kind of shows how we were able to, with experience over time, gradually get comfortable with the technology to allow it to be the gatekeeper for clinical use. So, I think everybody's familiar with the phases of drug development. There's clearly a host of opportunities here for applying these types of models in the drug development enterprise. You're probably all working on various contexts of use that pertain to some of these issues, whether it's preclinical or metabolism or other kinds of issues. I won't go too much into this slide, but suffice to say that there's a lot of opportunities, I think, to have complex in vitro models enhance our understanding of pharmacology and toxicology. York went over this a little bit this morning, so I won't belabor the point, but did want to emphasize that all of my experience, with precision medicine, and I would probably venture to say CDERS more broadly, has really been born out of the individual drug development programs and the review of those programs, and not necessarily a qualification effort. A lot of these biomarkers are drug specific. So that is a pathway that can be pursued where there is closer engagement with the agency through formal meetings, and you're able to discuss what the limitations of a technology are with respect to its application for a specific issue in a given drug development program. I'll talk a little bit about confirmatory evidence and some other opportunities for where these models could potentially fit. So the substantial evidence standard for FDA is what governs our ability to approve drugs, and we expect safety and effectiveness and adequate and well-controlled trials. Often in the setting of rare diseases, at least, that substantial evidence standard is met by one adequate and well-controlled trial plus confirmatory evidence. And confirmatory evidence can be mechanism of action, it can be observational data and natural history, it can be a number of different things. But what I think is probably important for this audience is that mechanistic and pharmacodynamic evidence are really instrumental in this substantial evidence pathway, as well as evidence from relevant animal models. And that's something that, I also review drug development programs for rare diseases, is critically important as we're discussing evidence of effectiveness. And just to illustrate, we often, have a disease that affects many systems in rare diseases. Excuse me. We give a drug intravenously. We're trying usually to measure response peripherally, but a lot of the diseases may affect the central nervous system. So we're often left with questions about what is happening, if you give a drug, specifically a protein therapeutic, peripherally, what is going on in the central nervous system? Is it having the intended benefits? And as you know, rare diseases, efficacy trials are often very difficult to conduct. So we're usually put in a situation where we're trying to understand whether a biomarker might be acceptable, whether it's in the periphery or the central nervous system, understanding what the toxic substance is, whether the biomarker reflects the pathogenic processes, whether the drug is getting to the affected tissues, and whether or not the toxic substance is being decreased by the drug in that tissue. So there's a lot of questions, and potentially this could be a very important area where these types of complex in vitro models could potentially benefit a great deal in the decision-making process. You're all familiar with the opportunities. I just laid this out just to think of a couple, more on the effectiveness side of the equation: confirmatory evidence of effectiveness, proof of concept for pediatric studies for the prospect of direct benefit. Pharmacological activity and dose estimation is a major issue that my office and the Office of Clinical Pharmacology deals with, and clearly metabolism and transport and those kinds of issues to help guide dosing and predict what might be the most effective dose. So to summarize, basically the trajectory that's been followed has gone from, concept to development to validation to optimization, implementation, and then we add on complications and expansion of the complexity of those programs. So the needs really drive the adoption, I think. The context of use, as we've heard several times today, as well as the risk, I would add, really guide the extent of validation that's needed. And these individual program specific applications, I think, are very instrumental in understanding how we might get to a path to qualification by understanding what the issues are, developing the standards, and understanding what the framework for review of those things is. So I'll close there.